Now let's go through Protocol 7, testing the different physical factors for microbial growth. So we will do 3 sets of experiments. We'll test a range of temperatures, a range of osmotic pressures, and a range of pH, and we'll try different organisms at each of those factors. So the first part of the experiment is testing a temperature range, and we'll do that with 3 different microbes. So we'll have a broth of each of these 3. We'll have a broth of Serratia marcescens, a broth of Bacillus styrothermophilus, and a broth of E. coli. We'll also use sterile swabs, and we have 4 TSA agar plates for this experiment. One of them that we'll put at 4 degrees, one of them we'll put at 20 degrees, one at 37, and one at 60 degrees. So we'll need a refrigerator for the 4 degrees. Room temperature, 20 degrees is room temperature. We'll need a 37-degree incubator and a 60-degree incubator. So for this temperature experiment, you'll first use a marker to draw 3 sections onto the plate and label those with each of the bacteria that we're going to test. And then label all 4 of those agar plates like that, because each is going to go at a different temperature: 4 degrees, 20 degrees, 37 degrees, and 60 degrees. And then you'd stir the broth and dip a sterile swab into the bacterial culture. And then using Sterile Technique and keeping the clamshell cover over the plate, you just swab it onto the correct section corresponding with the bacteria. And you'll repeat that for all 3 of the bacteria on that temperature plate. And then repeat it for each plate that goes at each temperature. The next part of the experiment is the osmotic pressure experiment. So for this, we'll need 2 different microbes. We need an E. coli broth and a Staph epidermis broth. And then you would have a set of sodium chloride plates of different salinities. So you would have one that has a 2% salinity, 5%, 10%, and 15%. And then you also have sterile swabs. So you're going to label each of those plates, each of the different salinities, in half. E. coli labeled on one side and Staph labeled on the other side. And then you would dip a new sterile swab into the E. coli. And using Sterile Technique, you'll swab that on the side of the plate that says E. coli. And then getting a fresh swab, you'll dip that into the Staph broth and swab that onto the other side of the plate where it's labeled Staph. And then that will get incubated at 37 degrees. And then the third part of the experiment is the pH experiment. So for that, we will need 3 broths. We need E. coli, Staph epidermis, and lactobacillus. And we need 1 TSA plate and 1 tomato juice agar plate. Now the difference between those 2 plates is the pH. The TSA plate, which is the lighter colored one, that is a neutral pH. And the tomato juice agar plate, which is the more reddish colored one, that is more acidic, so a lower pH. And again, you would label the plate with 3 different sections, one for each bacteria. And again, using fresh swab and Sterile Technique, you're going to put E. coli on the part of the plate where it's labeled E. coli, using Sterile Technique. and then a fresh swab. Repeat that with the staph on the section that's labeled for staph. And then finally, take the lactobacillus tube and swab that onto the section that's labeled for lactobacillus. So now you have all three bacteria on the tomato juice agar plate, and you would repeat that process with the TSA plate and then incubate those at 37 degrees.